first talk is going to be by Mike Stukel, who's going to talk to us about plankton size spectra, net primary production, and the biological carbon pump. So, Mike, over to you.
All right, can everyone see my screen and hear me?
Yep, we can see and hear you fine.
All right. I'm going to present some work looking at the relationships between plankton size spectra and net primary productivity and the biological carbon pump. And really, ultimately, the focus of this work is in trying to understand how different mechanistic relationships underpin the important roles that the marine ecosystem is playing for society. And this here is some work that we recently published in Global Biogeochemical Cycles. So if you want to see the nitty-gritty details of the paper, they can be found. But ultimately, we're looking at the role of plankton size in function for an ecosystem. And there are many reasons to believe that plankton size might be important in structuring planktonic ecosystems. And so it's worth considering some of the pressures that face organisms of different sizes. There are changes as you get larger in the surface area to volume ratio, which affects your ability to take up nutrients and also to diffuse oxygen into and waste out of your cells. Your size is going to relate strongly to your swimming speed because typically there's a relatively constant relationship of an organism swimming something like 1 body length per second. Particularly important to the biological carbon pump, settling velocity is strongly related to size, both in terms of theory from Stokes' law and from empirical relationships. Organisms' relationship to the water itself changes with size because of the impact of the Reynolds number and the strong relationship between Reynolds number and size. Essentially, if you're small, the ocean is viscous. If you're large, you're living in an inertial world. The amount of carbon, nitrogen, and phosphorus that you need to replicate is also, of course, going to vary with your size. The amount of cellular and intercellular organization you can have varies with size. And of course, who you can eat depends on your size, and who can eat you also depends on your size. And this is really important because of the large range of sizes that organisms exhibit in the planktonic realm. We've got everything from submicron size bacteria up to salps and jellyfish that may be over 100 millimeters in length. And just to put this in perspective, the range of sizes that we see in the planktonic realm is similar to the range in size from an amoeba to an elephant. So we obviously don't think that an amoeba and an elephant are going to face the same ecological pressures, and we don't think that an ecological community dominated by elephants is going to have the same impact as an ecological community dominated by amoebas. So all these give us good reasons to think about the relationships between plankton size and function. And a recent paper by Sarah Pompey et al., which also came out in GBC, actually developed a size-based model applying some of the basic ecological principles that we understand about how size affects organismal biology, and then scales that up into a biogeochemical model to look at the ultimate emergent properties of an ecosystem. And one thing that they found, amongst many others, that's particularly relevant to this study is that the PE ratio, as they call it in their study, or I might refer to it as the export ratio or export efficiency, correlates best with the exponent of the size spectrum and sea surface temperature. The PE ratio is really the proportion of net primary productivity that gets exported as sinking particles. And so basically, and the exponent of the size spectrum is basically the relative importance of large versus small particles. So really what this study suggested was that size of the planktonic community is the most relevant biological parameter in terms of determining whether you will have export driven or whether you will have a high export situation or a low export situation. And so we wanted to test this with observational data. And this is possible because my collaborators and I have basically been going out on cruises around the world for the past almost 2 decades now. And we've been collecting a similar suite of measurements in all these regions. And these measurements include, we use flow cytometry to measure the abundance of picoplankton. We use epifluorescence microscopy to measure the abundance and biomass of protists and phytoplankton. And we use size fractionated net tows to quantify the abundance and biomass of metazoa and zooplankton. And at the same time, all of this is conducted within the framework of Lagrangian experiments, where we're tracking a parcel of water for typically somewhere between 2 and 5 or 6 days, which gives us repeated sampling of a water parcel. And also on these Lagrangian arrays, we incubate experiments where we're measuring net primary productivity at 6 different depths. So we get an estimate of the vertically integrated primary productivity. And we also have a separate array out there that has sediment traps on it, so we can collect sinking particles. So we have simultaneous measurements over a period of roughly 4 days of the abundance and biomass of picoplankton, the abundance and biomass of nano and microplankton, and the abundance and biomass of mesozooplankton, as well as net primary production rates and sinking carbon export rates. And so now we can take these measurements and put them together to conduct what's known as a normalized biomass size spectrum. I'm going to be talking about normalized biomass size spectra a lot, so I want to just walk you through what this figure is. On the x-axis, we have size, and my size bins are split into approximately logarithmically spaced bins determined by the types of measurements that we're making. And on the y-axis, we have the biomass in that size bin in milligrams of carbon per meter cubed divided by the bin width. That's the normalized part of this biomass size spectrum. The normalization basically is done because that alleviates any issues you may have with potential different bin widths in your size bins, which may in some cases be experimentally constrained. So when we look at this figure, if we look at this point here, this point comes from our flow cytometry data. So that's the biomass of all of our heterotrophic bacteria, Prochlorococcus, Synechococcus, and picoeukaryotes summed together and then divided by the bin width, which here is 0.5 to 2 microns. So it's divided by 1.5 micron. The next point here is from our epifluorescence microscopy data. That's the biomass of 2 to 5 millimeter nanoplankton. And on as we get to the right side of this graph, we have size fractionated net tows, 0.2 to 0.5 millimeter zooplankton, 0.5 to 1, 1 to 2, 2 to 5, and greater than 5 millimeter zooplankton. And we have these consistent measurements across all those regions that I showed on the previous slide. And then we can fit a slope to this line. And this slope is giving us a crude estimate of the relative importance of large versus small phytoplankton. A very steep slope, so one that's more negative, means you're dominated by small organisms that rather than large ones. A shallow slope that's less negative means a relatively similar abundance of small and large organisms. We can also look at this in a little bit more granularity. We can look at the slope on the microbial portion of the size spectrum. We can look at the slope on the net plankton or mesozooplankton portion of the size spectrum. And we can also look at the biomass ratio. What's the ratio of net collected mesozooplankton to the microbial taxa, basically the phytoplankton and most of the protists. And so these are different metrics that we're coming up with to describe how size varies across regions. And this just shows you some of the size spectral variability across the region. So the plot on the left is showing you data from the Costa Rica Dome, the Gulf of Mexico, and the Southern Ocean subtropical front, which came from the Salt Poop study. So we can see some distinct variability between these studies. If we see in the Salt Poop study, there really were not many small nanoplankton. And we also see in some of these the evidence of large salps in the community. If we look at the Gulf of Mexico study, we see a substantial decrease in the larger size spectra, or larger microbes in the community. There really were not many of the microplankton in the community. And we can look at this, how this varies across the California current ecosystem and Hawaii Ocean time series as well. But rather than focus on individual stations and profiles, I want to look at what drives these size spectra. So in the graphs on this slide, on the y-axis on all these graphs, you have the slope of the NBSS, so the slope of that normalized biomass size spectrum. So as a reminder, Less negative values mean a relatively equal contribution of large and small organisms. Very negative values mean that it's really dominated by the small organisms. On the x-axis here, I have net plankton divided by microbial biomass. And so we see that the NBSS slope is really strongly correlated with the net plankton to microbial biomass. That's really what's driving the slope in our data set. When you have a high ratio of mesozooplankton to protists and bacteria, then you have a less negative slope. When you have a low ratio of mesozooplankton to protists and bacteria, then you have a very negative slope and you're dominated by small things. We also see a positive correlation, but quite a bit weaker, between the normalized biomass slope overall and the normalized biomass size spectrum slope for the net plankton portion as well. So this is telling us that when you have larger mesozooplankton, you tend to have a less steep slope, and that makes sense. If we look at the relationship between the NBSS slope overall and the NBSS slope of just the microbial portion, we find no relationship at all. So this is telling us that the size of the small phytoplankton, or the size of the phytoplankton portion of the size spectrum, is not driving the overall normalized biomass size spectrum slope. And that's largely related to the fact that when that slope tends to be more positive, when you tend to have more large phytoplankton, you tend to have more phytoplankton overall. So that has an offsetting impact on the NBSS slope overall. But now let's look at how the variability in the size spectrum relates to net primary productivity. So this shows the NBSS slope on the y-axis and net primary productivity of the phytoplankton community on the x-axis. And we see that there's a relatively strong correlation here. When you have a high productivity ecosystem, you tend to have a shallow slope. So in other words, when you have a productive ecosystem, it tends to be more dominated by the large taxa. When you have very low productivity system, it's dominated by the small taxa. We can also look at how net primary productivity relates to other portions of the size spectrum. So we see a very strong correlation between net primary productivity and the microbial portion of the size spectrum. That makes sense. We've for a long time known as a field that when you have high productivity, it tends to be dominated by large phytoplankton. We actually see no relationship between net primary productivity and the NBSS slope of the net plankton. So this is telling us that the size spectra of mesozooplankton is actually not tied to primary productivity, which was somewhat surprising, at least to me. And then we also find that there's a positive correlation between net primary productivity and the ratio of net plankton to microbial biomass. So that tells us that the ratio of mesozooplankton to phytoplankton tends to increase when your system is more productive, which makes sense because when you have a more productive system, you tend to have higher efficiency transfer to large metazoan zooplankton. When it's a low productivity system, you often have a lot of dissipation through the microbial loop. So now let's get at really the driving question behind this talk. What's the relationship between carbon export and some of these proxies? So first, I want to look at the relationship between export and net primary productivity, because that's one of the most basic things that we can look at. The exported carbon, of course, is produced by phytoplankton. So we expect that if there's more photosynthesis, you will have more export. And in fact, we do find that. We find a very strong correlation. Spearman's rank correlation is 0.51. And the slope of this type 2 linear regression is greater than 1, which importantly implies that the magnitude of the biological carbon pump increases faster than net primary productivity. So that suggests that the efficiency at which primary productivity is exported increases as primary productivity increases. But I will point out the caveat that these results are largely driven by the California current ecosystem and the Hawaii Ocean Time Series data. And the other data sets don't necessarily show that same relationship. So now, does sinking carbon export correlate with the slope of the size spectrum? So here I have the slope of the size spectrum on the x-axis, and we have export on the y-axis. And there is, in fact, a correlation, although that correlation is weaker than the correlation with net primary productivity. So this does tell us that there is higher export in regions that are dominated by large organisms, but that correlation is weaker than the correlation with primary productivity. What happens if we look at export efficiency? So export efficiency, again, is the proportion of net primary productivity that gets exported out of the euphotic zone. And it has essentially no relationship with a normalized biomass size spectrum. The Spearman's rank correlation was 0.14, the p-value is 0.21. So this tells us that size spectra are not statistically correlated with the efficiency of carbon export. So ecosystems with large organisms do not export a higher fraction of their NPP. They do have higher export, but that's largely because they also have higher primary productivity. We decided to also look at this in another way, so we used the statistical approach of lasso regression, or at least absolute shrinkage and selection operator regression, to predict log 10 export as a function of multiple different parameters. So for those of you not familiar with lasso regression, it's basically a multiple linear regression approach that tries to determine which variables are useful in predicting a particular functional response. So in this case, we have log 10 export as our response variable, and the predictors that we tested included the overall NBSS slope, the NBSS slope of just the microbial portion, the NBSS slope of the net plankton portion, the ratio of net plankton to microbial biomass, and then log 10 transformed net primary productivity, log 10 transformed surface chlorophyll, and surface temperature. We chose these latter three variables because those are three variables that, well, importantly, were measured in all of our studies, but also that are frequently used in estimating export patterns. So what we found when we used this approach is that none of our estimates of the size of organisms wound up being statistically significant. Rather, the lasso approach suggested that the variables we should look at are log 10 of net primary productivity, log 10 of surface chlorophyll, and temperature, with positive correlations to net primary productivity and surface chlorophyll, and a negative correlation with temperature, suggesting that export is higher in cold temperatures. So what's going on here? Why does size not predict export? Well, I have three potential things we should think about. First, maybe our analysis is just wrong. It's important to remember that the statement that the correlation is not statistically significant does not mean that there is no correlation. In fact, there was a weak positive correlation between export efficiency and the slope of the size spectrum. It just wasn't statistically significant. It's also worth thinking about the fact, as we look at the lasso regression, that maybe what we're seeing is really the fact that NPP, chlorophyll, and temperature are measured more accurately than our estimates of the normalized biomass size spectrum slope. And if we're measuring those more accurately, then it might make sense that those are the ones that are better working as predictors, simply because we have more uncertainty in the NBSS slope. So that's one thing that's always worth considering, although I don't actually think that's what's going on. Another distinct possibility is that there's a mismatch in timescales. Remember, the normalized biomass size spectrum slope is driven largely by the ratio of net plankton to microbial biomass. That was what we saw earlier in this talk. There's a very strong correlation that's telling us that it's really the ratio of mesozooplankton to phytoplankton and bacteria that determines that slope. But these organisms are responding to ocean variability with different timescales. Zooplankton have a roughly monthly timescale. Microbial timescales are on the order of days. NPP timescale, this should be, is approximately daily, and often driven by variability in light, which can be driven simply by clouds. And then the export timescale integrates over roughly the previous week plus of NPP. So it may be that when we're looking at these static snapshots, we're not seeing things integrated over the proper temporal scales to see the underlying drivers emerge. So that's another possibility that should be considered. And then the third possibility is that maybe size spectra are telling us more about food web efficiency than export. It's worth keeping in mind the fact that the idea of these size spectral slopes was originally developed as a steady state approach for looking at how efficiently energy gets transferred from small organisms to large organisms. And under that assumption, basically, when you're dominated by large organisms, you're going to have high trophic transfer to things like fish. When you're dominated by smaller organisms, you're not going to have efficient transfer up to higher trophic levels. And so if we think about energy going along one of two pathways, either to higher trophic levels like fish or being exported into the mesopelagic, maybe we're seeing a balance between the two. So maybe when the community is dominated by large organisms, we have food web transfer. And maybe when it's dominated by small organisms, that's because a lot of energy is being lost to the biological carbon pump. And that brings me to a little bit of a change of pace. I want to briefly present some unpublished results with my colleagues Moira Decima, Christian Fender, Andres Gutierrez Rodriguez, and Karen Self, looking at the impact of gelatinous filter feeders and how they can increase ecosystem efficiency. And this is really getting at an idea that's been hypothesized known as trophic amplification. And trophic amplification comes from ocean models that predict that future declines in phytoplankton biomass as a result of climate change will be amplified at higher trophic levels. So if we look at this graph here, this is showing for different climate scenarios the predicted decrease in net primary productivity in green, in light green, phytoplankton in dark green, zooplankton in yellow, and higher trophic levels, or fish, in blue. So what we see is that the fish decline substantially more than the phytoplankton. And this is caused in part by food chain elongation driven by a strengthened microbial loop. So as the ocean becomes warmer, we not only have a decrease in primary productivity, but that primary productivity shifts from large phytoplankton to small phytoplankton, and we essentially insert an extra protist and trophic step, and we dissipate energy. But it's important to keep in mind that these CMIP class ocean models that this is often based on do not explicitly simulate the microbial loop or food web reorganization due to climate change. And we think that this may be important because there's really substantial variability in predator to prey size ratio in pelagic organisms. This is a graph from one of my students' papers, Christian Fender. It shows on the x-axis, which is really the axis that's most important to look at, the predator to prey size ratio for a bunch of different types of organisms in black, and then for salps, which is what he measured in the study, in color. And we can look at some typical organisms. So Cnidarian jellyfish often feed at a predator-to-prey ratio of something like 1-to-1 or 10-to-1. Krill feed at a predator-to-prey ratio of around 100-to-1. Salps can feed at predator-to-prey size ratios of more like 1,000-to-1 or even 10,000-to-1. And to put that in perspective, a 1-to-1 predator-to-prey size ratio is a lion feeding on a gazelle. 100-to-1 predator-to-prey size ratio would be that lion feeding on a grasshopper. And a 10,000-to-1 predator to prey size ratio would be a lion feeding on an amoeba. So these are huge differences that may lead to differences in how food webs organize. These two graphs show you a conventional size structured ecosystem in which you start with some phytoplankton and you dissipate energy at each trophic level, and there's a strong correlation between trophic level and size. Big things eat smaller things with something like a 10-to-1 predator to prey size ratio. And the color shows the amount of energy. The graph on the left shows an ecosystem that starts with large phytoplankton. The graph on the right shows an ecosystem that starts with small phytoplankton. And if you start with small phytoplankton, you're at the same trophic level, you'll have smaller organisms. And so when you get to large organisms, you've lost much more of your energy. But if we think about organisms that actually feed at different predator-prey size ratios, we can have these compensatory food webs, where when you switch from dominance by large phytoplankton to dominance by small phytoplankton in a future ocean, you actually switch to organisms like salps that feed on a higher predator-to-prey size ratio. So that can lead to sustained high energy flux to things like this. This is a deep water Oreo that feeds on salps. And so even though you start with small phytoplankton, you may still have efficient energy transfer to larger organisms. So to study this, we conducted some Lagrangian process experiments at the Southern Ocean subtropical front, and we conducted 5 Lagrangian experiments. 3 of them were conducted in waters with salp blooms, and 2 were conducted in areas without salp blooms, so we could really identify the impact that these salps were having on the community. And for each of these Lagrangian experiments, we measured net primary production, size spectra, protist and grazing, mesozooplankton grazing, salp grazing, and trophic positions as estimated by compound-specific isotopic analysis of amino acids. And so if we look at that trophic position estimate, the graph on the left shows trophic position for zooplankton from 0.2 to 5 millimeters in size for each of our different Lagrangian experiments. So we see that those 0.2 to 5 millimeter zooplankton have trophic positions of about what we'd expect, falling between about 2.4 and 2.8 on the mean. So they're at a trophic level somewhere between an herbivore and a primary carnivore. And then we also looked at this for salps. And I want you to notice the difference here. This is 0.2 to 5 millimeter zooplankton. On the right, we have salps that feed at this really high predator-prey size ratio. These are organisms that range in size from about 10 millimeters up to 140 millimeters. And their trophic position on average was actually a little bit lower than that of the other zooplankton. So these salps were feeding on average at a trophic position of about 2.3 to 2.4. So this is really potentially efficient energy transfer to much larger organisms. And then if we put all of our different types of measurements together, we could estimate secondary production as a function of organism size during the salp blooms and non-salp blooms. So in these graphs, we have biomass production divided by the bin width. So this is biomass production per octave. So this is logarithmically spaced bins. So this is kind of like our normalized biomass size spectra, except this is now production instead of biomass. And so what we see, if you look here, the blue, red, and this blue were all in salp blooms. The orange and the greenish blue are in non-salp blooms. The productivity at the lower end of the size spectrum, the phytoplankton productivity, was very similar whether it was salps or not salps. But as we get to larger organisms, that high predator-prey size ratio of the salps allows us to transfer much more energy to the large organisms. So the presence of salps leads to somewhere between 1 and 2 orders of magnitude higher energy transfer to the large organisms that we fish out of the ocean, hence that many people care about. And we then compile data from some of the other regions that we've studied, some of the regions that I showed you in the first part of the graph. And we plot here the ecosystem transfer efficiency, which is the secondary production of greater than 200 millimeter organisms divided by net primary productivity as a function of surface chlorophyll, with color denoting the proportion of metazoan herbivory that's conducted by gelatinous filter feeders. And the takeaway from this graph is that alterations to herbivore communities are a better predictor of ecosystem transfer efficiency than primary producer dynamics. So now let me just quickly wrap up the talk. So from the first part of the talk, we looked at plankton size spectra and the biological carbon pump, and we found that carbon export is higher when large organisms dominate. However, this relationship results primarily from a positive correlation between NPP and export, and a separate positive correlation between NPP and size spectra, while size spectral slope was not actually a strong predictor on its own of export efficiency. And from the second portion of the talk, we looked at gelatinous filter feeders and ecosystem transfer efficiency, and we found that diversity in predator-to-prey size ratios provide the possibility for compensatory food webs, and ecosystem transfer efficiency may be as responsive to changing grazer communities as it is to changing phytoplankton communities, while compensatory food webs may offset declines in fisheries production predicted by the trophic amplification hypothesis. And of course, I'd like to thank many, many colleagues who have helped with all of this work.
Excellent. Thank you very much, Mike. Some great stuff there. We were a little pressed on time, but we could probably squeeze in one quick question if somebody's got one. Just sort of raise your hands if you'd like to ask a question. Let me just check whether I can see. So I can't say anything, so I will indulge myself and ask you a quick question. So you, in terms of the relationship between the slope and the export, you mentioned the fact that it might be related to the different timescales. Do you think you'd get a clearer signal if you looked primarily at regions that you might regard as being closer to steady state, sort of less seasonal areas?
I don't know that. I'm not sure to what extent looking at it at even places that we consider to be at steady state is necessarily the best approach. I mean, a lot of our data is actually dominated. Most of our data comes from the Hawaii Ocean Time Series, which is in theory one of these steady state systems, although it can vary quite a bit from things like eddies coming through. It's been shown that there's substantial high frequency variability driven by ocean circulation. What I think actually might be the better approach is to try and quantify this in more regions and look at regional averages and then compare, instead of having multiple points from within one region, look at the average for one region and compare this across something like 20 different regions. The problem is that everybody tends to use slightly different methodology. And once you're using slightly different methodology, that can drive differences in your size spectra. I'm actually working with the long-term ecological research groups at 4 different LTER sites. And we're trying to compare our size spectra, and figuring out how to make our size spectra comparable has been something that has consumed 2 different 3-day meetings so far. So it's a challenge. And I guess I'll leave it there so we have time for Raul to talk.
Yeah. Well, thank you very much. I saw there's one more question in the chat. So hopefully Mike can actually answer that on the chat and we can take on with the next talk. But thank you very much, Mike.